My name is Matthew Shair. I'm a professor in the Department of Chemistry and Chemical Biology at Harvard University. Well, there's enormous potential today to use chemistry to both study the basis of human disease and as a starting point for the development of novel therapeutics. Well, this is exactly what my group does. We use small molecules to probe the underlying basis of human diseases, including cancer, and then we use that information to develop new therapeutics. A key aspect of our research is the use of modern stereoselective synthesis to make molecules rich in stereochemistry and polycyclic structures that then can precisely target proteins involved in human diseases. So a specific project that we're studying in the lab involves two kinases, CDK8 and CDK19. Both of these kinases are involved in binding a large protein complex called the mediator, and the mediator is involved in regulating aspects of transcription. We inhibit these kinases specifically using a small molecule called cortistatin A, a molecule we synthesized in my lab. And what we found is that inhibiting these kinases is a new therapeutic approach to a blood cancer called AML, or acute myeloid leukemia. We then use cortistatin A and a variety of other biological techniques to study the underlying mechanism of how inhibiting these kinases leads to the growth arrest of AML cells. And what we found is that inhibiting these kinases leads to the upregulation of a series of key cell identity genes in AML cells. We're now using that information combined with our ability to synthesize potent and specific inhibitors of these kinases, and what we're hoping to do is start a human clinical trial in the near future targeting these kinases as a new therapeutic approach to treating AML in humans. The research in my lab is highly interdisciplinary. A unique aspect of my laboratory is that chemists and biologists are working side by side together. On the chemistry side, we use modern stereoselective synthesis to generate molecules that are rich in stereochemistry and polycyclic structures, and that enables us to make compounds that can target specific proteins. On the biological side, we use a variety of techniques. Most of them are genome and proteome-wide methods. We use CRISPR-Cas9 screening. We study iPS cells and primary patient samples. Students in the lab actually have the opportunity to study both chemistry and biology, as well as learn how modern therapeutics are developed. It's a very exciting time in the lab. We're gaining a greater understanding of the basis of human diseases like cancer, and at the same time, we're making small molecules that can make their way into human clinical trials to make a difference in patients' lives.